Folks, here we are on the northern coast of Maine, and I'm here today with Dr. Tony Reznicek, University of Michigan, Ann Arbor, and he is a Carex specialist. And Tony's going to go through, in this particular video, field characteristics of some of the more common Carex and field features which to identify them. So I'll just turn it over to Tony.
Well, we're standing in a very large sedge meadow dominated by several species of Carex, and if you look at it, you can see a large, uniform, fairly level stand of fine grass-like leaves. Those are the Carex, and if you look closely, you can see small differences in textures. You can see some coarse yellow-green leaves that stand out, and then a vast sea of really fine leaves, and intermingled in it will be tufts of other species. So the primary dominants here, and we'll see how to recognize them, are Carex stricta, which are the vast sweep of the meadow, very fine leaves, and often a darker green. And the yellow-green, broader-leaved plants are going to be Carex utriculata, and we'll look at a couple of the other species that are scattered through the meadow as well. The biggest problem that you have with Carex is that there is a whole lot of them, and somewhere around a little under 500 species in North America. Oh. So the big trick in learning them is, first of all, where to start, and the second big thing is breaking them down into smaller groups that you can begin to work with understanding them. And the basic way you do that is, first of all, starting to have a good grasp of sedge morphology, how sedges are put together. So I think that's where we'll start. That's your beginning. Now, worldwide, there are somewhere in the order of 2,000 species of Carex. And so one of the interesting things is when you get large genera of flowering plants that have species number in that order, normally what you have is a lot of variability. Some are annual, some are shrubs, some are trees, and so forth. And Carex is really unusual, and it's part of its difficulty in that all the species are herbaceous perennials, so the same life form throughout all the 2,000 species of Carex. So that gives you a uniformity that actually can be troubling to people at first, because they all look the same, and how do you start breaking it down? And you start breaking it down, first off, by having an understanding of the morphology. And one thing, of course, you look for right off the bat is the basic structure. And Carex have a great deal of uniformity in their structure as well, so that the defining feature of a Carex is a periginium. This is a bract-like structure that is tubular. It's formed into a bottle or flask or bag shaped structure, and it completely encloses the fruit, which is a very hard nutlet with only a tiny hole at the apex through which the style protrudes, and then the stigmas. So that's one of the defining features of Carex. And the other defining feature that people often note is that the flowers are unisexual. and that is fairly unusual in the sedge family. And I could back up actually to give a little bit of a definition of what a sedge is. I think that it suffices that it's more or less the wetland equivalent in a sense of grasses. They're grass-like plants with many of the characteristics of grasses and also rushes and a number of other plants. They're in the botanical order Poales, and while there are all sorts of rhymes about sedges have edges because of the triangular stems, only about 80% maybe of sedges actually have triangular stems, but essentially all of them have leaves in 3 ranks. And what that 3 ranks means is that in grasses, your leaves tend to be 180 degrees apart from each other. In sedges, if you look down on a sedge, you can see that those leaves are arrayed. If you just take 3 leaves as an example, they're arrayed at about 120 degrees from each other. So they are in ranks of 3 as you go up the stem, and it's easy to see that with most species. So back to Carex. When you're looking at a Carex, the way you technically identify them, if you don't know them, is to start working with the key, and this, of course, is where the frustration usually begins, because people who write keys, I often tell students that keys are written by people that don't need to use them for people that can't use them. And that's a terrible exaggeration. But of course, even for North America, where you have a little under 500 species, that is an incredibly complex structure of choices that you have to make to get down to a species. So back to my initial comment about actually starting to break Carex down into smaller pieces that you can recognize in the field easily, that is the beginning of learning to identify the species on site. And so you have a lot of field characters to go by, and the first thing you need to do is take an inflorescence and look at how the male and female flowers are arranged. And you'll remember that Carex are unisexual flowered. There are male flowers, there are female flowers, and in many Carex, those are arrayed in completely separate spikes so that the inflorescences have sexual specialization. So this one right here has these female flowers arranged in a long spike, and you remember all the floral parts are inside this bottle-shaped structure. And there's the 2 female spikes, and then up at the top here are the male spikes. So there is that inflorescence structure. You'll notice this has leafy bracts with the female spikes. That's another thing to take note of, and that is a beginning to line up the kinds of things you need to start identifying Carex. So that's one thing. The other thing, and the most dreadful thing in sedge keys when you're first starting out, is that you will read this very nice introduction that tells you that in order to use these keys, these complicated identification tools, you have to have mature, fully developed nutlets and perigynia. And of course, the first thing it asks you is how many stigmas do the flowers have? Well, they've fallen off by the time the flowers have matured. So, well, key writers aren't sadists. Well, many of them aren't. And so, obviously, there are things they know about looking at things that that you don't. And one of the things is that there's a strong correlation, with some exceptions, between the shape of the inflorescence and the number of stigmas in the flowers. So something like this, with an elongate inflorescence, sexual specialization in the spikes, female spikes, male spikes, leafy bracts, a plant that looks like this will have, with some exceptions that you must learn, 3 stigmas. And we'll look in a little bit at the distinctive morphology of an inflorescence of Carex that is characterized by having 2 stigmas. We can do that relatively quickly.
You just talked about a correlation between the overall arrangement between 2 and 3 stigmas. Is there one between the shapes of the nutlet and the stigmas?
Yes, absolutely. In those species of Carex where the stigma number is 3, the nutlets are triangular. And in those species where the stigma number is 2, then the shape of the nutlet is lentil shaped. And that's flattened and disc-shaped with some variation and modification. So that helps too. You have to open up this periginium, this little sac that's characteristic of the whole genus. And of course, sometimes that is a little difficult because they're tiny. There is no broad correlation between the shape of the periginium and the number of stigmas, because some things with 2 stigmas have plump, round perigynia, so you have to be careful there. You have to actually dig in, open it all up, and get a look at the nutlet. And I can show you a little bit about what the nutlet looks like by opening up one of these, and we can have a look at that at some point as well. Okay, so I mentioned before that that there was a correlation between inflorescence form and the number of stigmas. And so here we have a species that is quite different in construction of the inflorescence from the one we were just looking at. And you'll notice 2 things here. One is that the inflorescence is much shorter and smaller. And the second thing is that the spikes, the units of the inflorescence, they're not really spikes, we'll colloquially call them that, are short, and they all look the same. If you took this bottom one here and pulled it up and stuck it up near the top, it would basically look the same. So there is no sexual specialization in the spikes. There aren't really conspicuous male and female spikes that are highly differentiated, and so that inflorescence form is characteristic of species with 2 stigmas. So with a little bit of practice and a little bit of learning the exceptions, you can actually drive by a sedge and say, oh, that has 2 stigmas, that has 3 stigmas. This is actually really useful because if you are still keying them, this cuts your keying in half right there, and you don't have to dig around with a dissecting scope to figure it out. You don't have to open up the periginium, so it's a very handy thing. Again, you do have to learn some exceptions, but as a general rule, it actually works really well. So I want to cover a few more things that are linked to the identification of species using this as an example before we actually start to look at some of the common wetland species and work on their identifications. One of the things that you have when you have an inflorescence like this where all the spikes are the same, of course there are male and female flowers in every spike. Otherwise they wouldn't be essentially identical. So the next thing you have to find out, and you will be asked this in keys, is where the male flowers are. And that's a very important thing, because you remember, even though these spikes all look identical, all flowers in Carex are unisexual, either male and female. So there have to be male flowers in here. Where are they? And the trick to looking for them is really twofold. First, always check the apical spike, the uppermost one in the inflorescence. And the reason for that is that often in that uppermost spike you have more male flowers than in any other spike, and therefore they are more conspicuous, because what you're looking for is empty scales. You remember, these are nearly mature fruits, so flowering took place quite a long time ago. And if you're a male flower in the plant world, you have a sad fate. Once you've shed your pollen, you're out of there. So the male flowers just basically disappear. But you can look at this and you can see that that terminal inflorescence has below those things sticking out, right, which are the perigynia, a number of
Very
scales
small but obvious.
Are apparently empty. Well, that's where the male flowers were. And the other trick is that if you hunt around and you cannot find any male flowers, then that is a good sign that they were the reverse arrangement. That is, female flowers below and male flowers above. Why? Because perigynia are tiny when they are flowering, and they expand and grow. If there's a few male flowers at the apex of the spike, the perigynia just below them will grow and enlarge, and they'll conceal where those male flowers were. So if you can't find male flowers, say that they were apical in the spikes, and you'll be right enough of the time to be satisfying. There will be times you'll be fooled, but it will work fairly well. So those are two of the basic things that often frustrate people tremendously when they're first starting to get to know sedges. One is how many stigmas there are, because they've all broken off when the perigynia are mature, and you're picking away at it with the needles under a dissecting scope, and every time you try and open one up, it springs off the table and onto the floor. And this is not the way to do it. There are ways of learning that. And the other is where the male flowers are. If you master those two items, you are going a long way towards actually getting the answer to the sedge you're looking for. Now, of course, I'm talking a little bit about keying, but in fact, those features are all useful when you're looking towards field recognition of some of the major groups, because, of course, the two inflorescence types you have here are representative of the two largest groups of Carex. This one, botanically, is subgenus Carex, with separate male and female flowers, big long inflorescences, long spikes, leafy bracts, and 3 stigmas, generally, with some exceptions. And this is botanically known as subgenus Vignea, a weird name, dating back to 18th century Latin. And this is the group that has 2 stigmas, almost always, uniform spikes. All of the spikes are identical, no differentiation. So that's a major element, recognizing those 2 big groups, whether you're zeroing in on field recognition or whether you're going to spend a comfortable evening with a sedge key.
Comfortable? You sure?
Of course.
Oh, I forgot. Yeah, it's enjoyable too.
By a margarita, maybe, you know, that always makes keying go more smoothly. Okay, let's start with looking at what this plant is. And we have a number of features on this lovely plant that enable us to identify it clearly. This is one of the most important species of sedge meadows across the temperate, boreal, and Arctic parts of the globe, and this is Carex utriculata. That it is one of the bladder sedges, and those are sedges where the periginium is large. It's often yellowish in color, and the body of the periginium is inflated into air-filled sac. And you can already guess that since this dominates wet meadows, that this is actually a species whose perigynia are adapted, at least in part, to floating on the surface of water, and it uses that to get dispersed, whether directly or whether by being presented to waterfowl, which then eat it and then disperse it. Here you can see Carex utriculata with bladdery, inflated, air-filled, yellowish perigynia in very long spikes, and that is also diagnostic of this particular species. And the perigynia are densely packed and form really a thick spike visible at a considerable distance. And here are perigynia pulled out of the spikes, and you can see relative to other Carex perigynia, these are large and plump. This plumpness is because they are filled with air, and that nutlet is just loose inside it, not densely wrapped by the periginium. And this is basically an air bladder or a small balloon, air-filled balloon. And it has a couple of other features. If you see this in meadows, you will see expanses of it, and that kind of tells you that it is in fact a rhizomatous species. And I pulled this up and was successfully able to get this long horizontal rhizome. This is a rhizome from last year. So you can see immediately how this forms these colonies. And it also has large, conspicuously spongy, soft bases. That also is a feature that's very distinctive for this plant. The inflorescences are always associated with these large spongy bases, and you can see them, the old leaves at the bottom of this flowering shoot. So that feature, the bladder-like perigynia, which you can colloquially call fruits. Of course, the fruit is actually the nutlet inside, yellowish color, and long creeping rhizomes that form dense colonies. So this is one of the most important species of wetlands actually across all of North America. In fact, all of the northern part of the globe. In mountainous regions, it will extend well to the south. In the Rockies, it actually extends down the spine of the Rockies, all the way actually into central Mexico. And even in the eastern mountains, it extends all the way into the mountains of North Carolina. So this is one of the most wide-ranging and most important wetland species worldwide, Carex utriculata. There are a few other similar species. Most of them do not have these massive, spongy old leaf bases, nor these long creeping rhizomes. The entire plant color here is kind of a yellowish green, and so these subtleties of color are also something that will enable you to identify sedges with some practice and learning the different colors as you see them arrayed in large clones, typically in meadows, from their rhizomatous growth. Now this species, the example that we used for the species group that has two styles and with the very different inflorescence, lacking the sexual specialization of spikes, all spikes being identical in this, spikes in quotation marks, is another circumpolar wetland plant. Circumpolar means they're in boreal and Arctic regions across the northern regions of the globe. And this also is a major species of wetlands. It is identifiable by a couple of features that are fairly easy to see from a live plant. First of all, of course, you've got that inflorescence of uniform spikes. And secondly, if you look at the leaf color, you can see that it is actually a blue-green color or a gray-green color. And that feature, along with the clumping habit, this species does not have creeping rhizomes. Every plant you see is a separate individual. Also helps to identify it. And if you have a hand lens, what you can do is take off these perigynia, and you will see two features of the perigynia that will help identify it. One of them is that the edges of these perigynia are rounded and blunt and not at all sharp-edged. And the other is that the perigynia are densely covered with little tiny warts called papillae. And these papillae probably function similar to the bladdery fruits of some of the big bladder sedges in trapping air and acting as flotation devices, because this is also water dispersed. If you're in a wet area, you'll see also the perigynia this floating in the water. So the combination of gray-green foliage, papillose perigynia, and blunt-edged perigynia will be very helpful in narrowing this down to Carex canescens. Carex canescens, among all the species that look like it, have more perigynia in these spikes than any other. Most of the plants that look very much like this have only a few perigynia, typically less than 10, and Carex canescens, especially on the lower spikes, has more than 10 or 12, often 15 or 20, and those numerous tiny papillose, gray-green, blunt-edged perigynia are diagnostic for this plant. And again, very important in peatlands throughout the northern part of the globe. So this is another one of those species that's extremely widespread. Again, in the mountains southward to the southern US, and also southward in the eastern mountains, and very widespread in Asia. Again, in open wetlands, perhaps slightly more shade tolerant than the Carex utriculata, the big thing that we see dominating so many of these meadows. So there is how you can start beginning to break down these characters and identify things. Here is the upper part of the inflorescence of Carex canescens, and you can see the terminal spike, the uppermost spike in that inflorescence right at the top of it, has a very conspicuous zone of scales that don't appear to be plumped up and bulged out with perigynia, and it is those scales that mark where the male flowers were. So this has the male flowers below the female in the inflorescence. And that's quite a distinctive feature of these plants. This particular species needs a little more explanation about its characteristics than some, because we have to introduce a couple of concepts. One of them is that this, of course, when you look at the inflorescence, there is the male spike at the top. There is the female spike. In between, there are some in-between spikes with the apical portion male and the lower portion female with perigynia. Well, that happens. That's not at all uncommon. Maleness and femaleness in sedges is a hormonal gradient, and so you do get on the apex of many primarily female spikes a few male flowers. And this is probably functional also in more effective pollination. Now, if you look at this inflorescence, you would, with this leafy bract, you'd be very correct in saying that this belongs into the subgenus Carex, which is characterized by 3 stigmas coming out of that little opening in that perigynium. But this happens to be one of the exceptions that you have to learn. This is Carex stricta, and it's one of the most important wetland species in eastern North America. It was probably, with Canada blue joint, Calamagrostis canadensis, one of the important species in the beaver meadow cycle in eastern North America and that kind of wetland habitat, and is an extremely critical wetland plant that many people will be intimately familiar with, sometimes with their entire body length, because this is the species called tussock sedge, and anybody who's been in wetlands will have walked through tussocks of this plant, which are raised columns of root masses and rhizome masses topped with leafy growth. And these tussocks are actually one of the diagnostic features that you look for in Carex stricta. So if you're walking through a meadow and you have no idea whether it's better to try hopping from tussock top to tussock top and falling in, or just simply going in by stepping in between them, then you have already gone a long way towards identifying Carex stricta. And Carex stricta forms great stands by elongate rhizomes, and it also has, besides just those elongated horizontal rhizomes, it also has vertical rhizomes. And tussock formation, whether it happens in Carex stricta or not, and in some meadows it happens and in some it doesn't, seems to be linked to the differential growth of horizontal versus vertical rhizomes. And it's thought to be linked to water level fluctuations, and again that is of course then linked to beaver meadows. So this species is a very intricate plant that is actually in many wetlands throughout eastern North America, from Canada all the way down to Texas and Mississippi. So how else do we recognize Carex stricta? That actually requires a couple other characters, and one of them is that if you pull apart a flowering stem, you can look very closely at the sheaths, and you can see that there are strange looking fibers on that sheath that look for all the world like ladders. And this is what you see in descriptions as ladder fibrillose. And they really do look like ladders. And Carex stricta is one of the species that has that developed to a very great extent. And also among similar species that look like Carex stricta, you have to look at the little leafy bract. And in Carex stricta, this little leafy bract is just about the same length as the inflorescence. In this one that I've picked, it's maybe a centimeter longer or a little more, but basically the same length. In many other species that are similar, they either do not have these ladder-like fibers, or this bract is much longer. So you can identify Carex stricta not only by flopping from tussock to tussock, but also by the process of looking at these technical features of the bract inflorescence proportion and whether the sheaths at the base of the plant have the ladder-like fibers. So if you look closely, you can see that ladder-like structure, which consists of horizontal fibers. Those are sclerenchyma fibers that are associated with vein tissue. They probably have no veins there, no vascular tissue, just the fibers. And when the sheaths mature, the little filmy tissue in between disintegrates and the veins tear from the center and leave these lateral fibers connecting the edge of the leaf to the main set of veins in the middle. And this is a process of disintegration when the plant grows and expands in thickness, but it's very characteristic in many species of Carex and very helpful in diagnosing some of the species. And it's a structure that's actually fairly hard to explain. You just have to actually see it. And here you can see it freshly breaking on a fairly young leaf, and it results in this very striking structure that's visible, especially in this plant, Carex stricta. The only time you run into any problems seeing it is sometimes when the plants grow in deep water and the veins don't dry and peel apart quite in the same way. But this is very striking as a diagnostic feature for Carex stricta. So here we have another one of the really, really important wetland dominants that actually grows in really wet places and dominates vast tracts of wetlands in the boreal zone, especially across the entire globe. And this one here, you can see immediately the inflorescence characters of sexual specialization in the spikes, male spikes above and female below, conspicuous bracts associated with the spikes, and so that tells you it's subgenus Carex. This is not one of the exceptions, so this has 3 stigmas, and it has a couple of really distinctive features that allow its ready identification, and it's very nice because this is such an important plant. First of all, the perigynia, those sacs that enclose the nutlets, are hairy. And if a sedge has hair on it, it is a get-out-of-jail-free card. You can utilize that to short-circuit identifications, eliminating hundreds of species. So that's very handy. And the second thing, the leaves on many plants of low nutrient habitats are very narrow, enrolled, and they are what is called filiform involute. That's a nice technical term for a leaf that looks in cross-section somewhat like an old-fashioned eaves trough. And not only that, but these leaves are very, very narrowed as you approach the tip, and they take absolutely forever to go about the business of coming to an end. And you have these long hair-like tips. These long hair-like tips actually curl sort of like a pig's tail late in the season and turn white because the tissue dries, and you can actually identify large stands of this species, Carex lasioscarpa, from an airplane because it produces these great white sheets over the peatlands, always in the very wettest parts. It is colonial from long creeping rhizomes, and you can see the rhizome there. This one is coming up to the surface. That's probably in response to the beaver raising the water level. But they spread to form large colonies, and whenever you find it, it's typically on the leading edge going out into the water. So you have to get wet to see this species, but it has a very important role to play ecologically because it is the species that is instrumental in forming a root mat advancing out into water, and so is a great mat former. And we can have a look at some of the characteristics up close, in particular the leaf tips and the perigynia. So here, if you look close, you can see those plump perigynia, and they are completely coated with a felting of fine hairs. And that's one of the diagnostic features, in fact, combined with those filiform involute leaves that taper to a long point. That's absolutely diagnostic for Carex lasioscarpa. So here you can see the very, very delicate, fine leaf tips, which mark Carex lasioscarpa. They taper almost to nothing, to a very delicate hair-like point that actually becomes curly later in the season and whitish, and that whitish color in masses of millions and millions of leaves produces a distinctive and striking look to the meadows of Carex lasioscarpa. This is a species which dominates square miles, probably thousands of square miles, in the boreal zone in the northern hemisphere. A wetland species that is exclusively eastern North American is Carex lurida, here with these very pale yellowish pistillate spikes. You can see the staminate spike and the leafy bract. And Carex lurida is one of those very widespread species that follows a slightly different pattern of many wetland species, in that it is not a circumpolar species. It doesn't circle the globe, but rather is eastern North American and extends all the way down into the neotropics. It occurs in northern South America, mountains in the Caribbean and Mexico. So it's quite a different pattern. Carex lurida also is a dense clump forming species, which you can't really see from this one stalk, but it forms a dense clump, sometimes of many, many stems, and has a very compact inflorescence of spikes that have essentially no stalk whatsoever. They are sessile, and so the inflorescence is very compact. Other similar plants generally have the lowest spike in the inflorescence, often nodding, on a separate peduncle, and usually darker green in color. These are also very soft, very inflated, and very easily compressed, because if you look at these under the microscope, you'll see that they have very few veins, only about 8 to 12 veins through the whole perigynia, and so they're not heavily reinforced, and very soft, very easily compressed, but still bladder-like and obviously very dispersible given the enormous range that it has. So this is a clump-forming species with bladder perigynia, very soft and with little reinforcement. And as a rule, it prefers acidic soils. So it has another ecological restriction besides being Eastern North American. You can see more closely the tightly packed bladdery perigynia, and you can also see at this distance that the tapered end is really quite long. That tapered end, when it's present, is called a beak, and beaks are often very important in the systematics of sedges and in working through the keys, and this is one of the ones with the longest beaks and most easily seen and distinctive beaks, so that illustrates a nice structure. But Carex lurida, tight inflorescence of sessile spikes, pale, soft, and obviously, again, dispersed by water and occurring in wet places. With this species, which, if you look at the inflorescence, you can see that there's no specialization of male and female spikes, and it's very compact, and it lacks leafy bracts. So you know this is a species with 2 stigmas and is a member of subgenus Vignea. But with this species, we look at one additional feature that you see in a number of relatively common wetland species, and that is the inflorescence branches, which in many species are just one single unit, one spike, is actually a branched structure with multiple axes, each with their own staminate apex and pistillate portion. So this is Carex stipata. It's one of the very commonest wetland species, transcontinental. It reappears in parts of Eurasia as well. Not exactly circumboreal, but very widespread. Carex stipata exemplifies species of subgenus Vignea with compounded inflorescences. And those actually help you a little bit, because all species in North America with compounded inflorescences have the staminate flowers apically, so you don't need to hunt for them, and they're very hard to find because they're small, and the perigynia in this are quite large. Carex stipata has a couple of very distinctive features that allow it to be readily identified. First off, the perigynia are extremely long and narrow and shaped like an old shoemaker's awl, which is a very narrow, sharply triangular point for digging in leather. And also the stem, which is often termed a culm in grasses and sedges, C-U-L-M, is very broad, very sharply triangular, almost wing-edged, and soft and easily compressed, so that if you squeeze it, you very quickly can crush it, and if you squeeze hard, you cannot roll it between your fingers because you flattened it. It's not wiry, it can't be rolled. And that broad, sharply triangular stem is very distinctive for Carex stipata and a few other relatives, and to distinguish stipata precisely, you have to look at the sheaths, and the sheaths have a very distinctive feature also in having very delicate cross corrugations on the sheath. If you look at the very tip of the inflorescence of Carex stipata, you can see the perigynia pointing upwards. They're not quite mature, they haven't spread out as much as they will, but inside that very apex, where the perigynia beaks go beyond the tip, you can see a little tiny nubbin with a little remnant, perhaps visible, of stamen, and that is the staminate flowers at the apex of these inflorescences, of every single inflorescence unit in Carex stipata. Here you can see the very long, pointed, awl-shaped perigynia with a very distinct beak, and you can also see that the base of the perigynia is whitish. And members of subgenus Vignia, those Carex with short, stubby inflorescences that are with all the spikes bisexual and 2 stigmas, don't develop bladdery perigynia, but rather they have whitish styrofoam-like tissue inside the perigynia that presumably acts as a float. One of the commoner species of acid soil wetlands, peatlands, and mineral soil wetlands across the northern hemisphere is Carex echinata. Carex echinata has the common name of star sedge. You can see the very striking star-like pattern of the perigynia of Carex echinata when they spread and even reflects at full maturity. And you can also see a couple of other features that are important. One is the relatively long and sharply pointed beak, and the other is that this is one of those species with the male flowers below the female flowers in the spikes, and you can see that small area right below the terminal spike where the staminate scales are, and those scales are empty, and that's where the male flowers were. So that, and Carex echinata also has sharp edged perigynia. The perigynia do not have wings, but the edge is relatively sharp. It's not bluntly rounded, although the actual margin is blunt, but the edge is narrow. So that kind of intermediate structure with those reflexed perigynia clearly mark Carex echinata. Very sharp, pointed perigynia and relatively long, over 3 millimeters long. That's relatively long for a Carex.
I would like to discuss and ask questions about the taxonomy, the systematics, the ecology, and some of the field habitats of some of these species. And so this will just be a general discussion, a fireside chat without a fire. So maybe I'll start out with there were in a long history of classifications of the genus Carex, and now with the modern phylogenetics and relationships, if you could sort of explain maybe how the morphological matches the classical arrangements, or if there's obvious groups and non-obvious groups, or however you want to present that?
Well, as a general rule, I think there's a reasonably close match. Carex is a natural genus, that is, it's a evolutionary unit. It's not a conglomerate of unrelated things. To make it fully coherent and cohesive, you actually have to add into it a few satellite genera with a small number of species, very few of which concern anything with North America. You have to add in Cobresia, Uncinia, Schoenoziphium, and a couple of other small satellite genera that have been split off in the past. In terms of evolutionary breakdown of the genus, the two large subgenera, Carex and Vignia, which Carex is primarily three-styled and Vignia is primarily two-styled. They have very different inflorescence. Carex typically has sexual specialization in the inflorescences with male and female spikes, or sometimes some other specializations like bisexual and female, and Vignias have almost totally uniformly bisexual spikes. Those groups are well supported, they're uniform groups, they're good evolutionary units. The interesting thing is that the basal groups in Carex actually appear to be subtropical woodland things from Southeast Asia with great paniculate inflorescences, highly branched inflorescences. That's rather interesting. That was unexpected. And we were able to resolve one of the great mysteries in the study of Carex evolution, and that is the relationships of those species whose inflorescences were so highly reduced and streamlined as to become just single androgynous spikes, androgynous being female flowers below and male flowers above. Those appear to be related more closely to the genera like Cobresia and like Uncinia, and hence that's one of the reasons why those genera had to be put into Carex.
Where does it sit inside the family?
It sits actually very tightly with Scirpus and several of the close relatives of Scirpus. That's Scirpus in the strict sense, the modern sense. And the linkage between Scirpus and Carex is easy to see, actually, because that little periginium in Carex, that little sac or flask-like structure or bag-like structure, that is actually a profil. That's the derivation of the periginium. And a profil is really just the specialized first leaf on a branch. And so if you look, for example, at a Scirpus inflorescence, each branch, and of course you can really see it clearly on the lower branches, each branch comes out of a small tube. And that tube is the homologue of the Carex perigynia. That's what the Carex perigynia originated from. So you can think of the Carex perigynium as being an entire inflorescence branch pulled down inside that tube and closed off and reduced to a single female flower. That's what has happened in Carex, and there are even genera close to Scirpus that have those profils with a female flower, so they can show directly how you derive Carex, and that is the little-known Asian genus Sumatroscirpus, which actually ranges from southern China all the way into the tropics. So that shows the link between Carex and the rest of the Cyperaceae, and Scirpus is its and allied genera are the closest relatives.
So some families, monocots or dicots, are dominated by wetland plants, and thus some genera are like Potamogeton, for instance, is obviously a wetland aquatic genus, but Carex, are all Carex wetland plants, or?
Well, that's a very interesting point that links to the evolution of the Periginium, because it's thought by some people that the sedges essentially originated as the wetland equivalent of grasses. They originated, as did the grasses, in the Gondwana subcontinent, the giant continent of Gondwanaland. And a long time ago, 100 million years ago, roughly maybe, and all sedges are characterized by having an extremely hard, extremely thick-walled, heavily silicified, bony nutlet as the fruit. That's very different from the fruit of grasses, which is a leathery, walled grain. And that is so uniform in the entire family that it's thought to be deeply embedded in the genetic constitution, channelized in the genome. And so it appears to be very hard to modify that nutlet. And that's why we see that it's uniform throughout the entire family. And so this is kind of limiting, because it probably evolved to survive the ingestion by waterfowl, or possibly even aquatic dinosaurs, because it dates back that far. So sedges in general are wetland plants, with some exceptions in the tropics, because obviously if you're dispersed by waterfowl, what are you going to do in a forest,
Right,
or a prairie? And if you have a difficult time modifying that hard, highly conserved nutlet, you're stuck. So Carex has simply sidestepped the problem completely by leaving the nutlet as it is, putting it inside the periginium, which is just a bract. It's a modified leaf, a profil, and then you can modify that bract to any dispersal mechanism that you want. And many Carex are ant dispersed, and that's obviously a good thing for forests. Many Carex are dispersed in the fur of animals or by blue jeans. Many Carex are dispersed in mud, on birds' feet. They cling. So there are even sedges, Carex, that have balloons as dispersals. They're wind dispersed. So they're using the periginium as the dispersal organ suddenly opens up the entire panorama of habitats for Carex. So many Carex are upland, many of them are forest understories. In fact, eastern North America has as one of its unique floristic features the fact that Carex has diversified and radiated extensively into the understories of the eastern deciduous forest. That's one of the unique features worldwide of the deciduous forest flora. And there are several groups that have speciated extensively in forest understories. So the tale of the evolution of the periginium as a dispersal unit and the diversification of Carex into non-wetland habitats is tightly linked.
I wasn't aware of that because in the West there are several forests, but not many. But there are several forest species in a conifer forest.
Yes.
But more so in the East. I hadn't realized that, and that's unique worldwide. Then
Yes.
I didn't realize that. Um, and uh, I guess when you have a wetland species, is it highly restricted, do you think, to a wetland, or are they variable? They can be in water. How ecologically do you think they're responding to that water? Or is it more of a restriction that, um, you know, restricted to wetland or eliminated from uplands? If I'm getting it across, logically.
Yes, the primary wetland dominants are obligate wetland species, but there's a large group of species that where water does not seem to be the controlling element of their ecology. For example, many of the species in the notorious section ovalis occur over a broad moisture spectrum, but they are seed bankers of disturbed soils, and they will come up on the bottoms of beaver ponds when the beaver pond goes out. They will come up when a forest is disturbed and there may be a wet depression, there may not, they may still be there. Some species range in their ecology all the way from dry gravel flats to logs floating in beaver ponds, and it's the bare soil or bare sunny habitat that is the determining factor.
Within the genus, what percentage might be wetland plants and what percent might be upland plants?
Wow, I think a lot of sedges are facultative because they clue into factors other than moisture. Factors other than moisture are more important to them, especially some of the seed bankers, which have specific germination requirements and disturbance requirements. But I would say that there are relatively few, probably only 10% of the genus, are really large-scale wetland dominants that are community forming primary habitat dominants. There's quite a large number, maybe half the genus, that are wetland plants but very local kinds of species, small species of specialized habitats in bogs and fens and things, but rarely, they don't dominate large tracts. So that would be a large chunk of the genus. A number of species, a modest number that form the vast numbers of square miles of sedge meadows and tundra and alpine meadows in the wet soils. A large number of species that are wetland but very local. A modest number that are facultative, they react to things other than wetland. And then a small number that are really dryland, upland species that are of prairies and even semi-deserts and so forth, dry rocky slopes. And then a substantial number also, gosh, in North America, it's probably 25% of the genus that are woodland, and many of them are swamp forest or wet mesic forest plants, and they range all the way up the spectrum to dry forest plants. But they really are more forest plants than they're forest plants first and wetland plants second. And then you get some that are flexible about that too, that seem to be shade tolerant but if they can grow in the open, they'd love it. And those are probably a small proportion. So there's quite a diversity in a range of categories, especially specialized wetland things that divide the habitat up very finely in peatlands in particular, which is where there's a great diversity, especially in fens, where you can get a high diversity of sedges, very local in their ecological, in their distribution, both geographically and ecologically.
I have another question that we've had where, as you're aware, with the National Wetland Plant List, we're going through an update right now, and one of the plants that we downloaded herbarium data, and we got, of the 1,400 records, many were in wetlands, but then we had all these upland records, and so we're having disagreement about, some people think it should be an obligate or fact wet, other people this. My experience is I see it in dry abandoned meadows, different places. What is how would you look at Carex vulpinoidea as a wetland species?
Well, it is, I think, a species that is early successional, and so it's responding to many other habitat cues other than water. And I think it's a facultative wetland plant, and I'm not exactly sure offhand. I'd have to think about it, but you could probably look at herbarium specimens and see what proportion, say, they come from wetlands and what proportion they come from uplands, and that would help you divide that out. That's probably the approach to take, but that's one of those species where I believe it's responding to other environmental conditions besides water supply.
Okay. And then diversity. Are there parts of the North America with higher diversity of Carex and endemism? Are there clusters of those?
Yes. The southeastern U.S. has a lot of local and regional endemism, especially to the coastal plain province, but also to some of the interior areas, the two mountain regions, the Appalachians and the Ozark, Ouachita, Boston Mountains of Arkansas, southern Missouri, and eastern Oklahoma. Those areas both have a number of endemics, some very local indeed. So eastern North America is a high point for Carex diversity, with endemism highest in the southeast. The Great Plains are a low point. There are Carex there in the plains and the prairies, but diversity is not high and endemism is essentially zero. And in the west, there is a fair bit of mountain endemism, especially in California and the Coast Ranges.
Okay. And like the Rockies, a lot of well, at least what I think or my experience is, there's Carex in the high country that are, you see when you go to Alaska.
Yes.
That have been moved with glacial activities and distributions.
Yeah, there are many high Arctic species that come down the mountains. Many reach as far south as Colorado. A few go even further south. Essentially, none reach Mexico, but there are even more species in places like, especially Montana, and so there are a lot of Arctic species that reach their southern limit in the high areas of tundra in Colorado. Colorado has by far the most alpine tundra of any of the lower 48, I believe, and that's where the diversity of tundra species is highest, and they doubtless are survivors of past colder climates.
So here I am. I bump into Tony Reznicek at the hot dog stand. And I say, Tony, I want to be a reasonable field expert in the genus of Carex. How much time is it going to take me to be reasonably proficient at a lot of these more common wetland sedges for my job?
I think it's easy to do in a summer with a little application. I think that it's really helpful to get an introduction to Carex, especially a little bit about the morphology and the characteristics. It's really helpful to see them in the field, so after you get that introduction, um, look at them in the field. Also, get a key for your region. There are good Carex keys for much of North America, and start running some things through the key. Start running some species you know through the key, so that you understand the structure and layout of the backbone of the key, and that way you'll be able to make decisions more quickly when you meet with species that you don't know, but you will know which groups they belong to. So a summer should get you easily familiar with most of the common species. There are some really hard groups that will take a lot of work uh, to get familiar with, and there are some groups that have a lot of really uncommon species that you don't see very much.
Mm-hmm.
And it takes a while to see enough of them that you can look at them and say, ah, yes, that is that rare species, which I've seen only 6 times in my life, but I know it now.
Right.
And so, but in a summer, you can develop facility with all of the common species. And interestingly enough, although those Carex keys look incredibly imposing, and you're going through couplet after couplet with measurements of tenths of millimeters and a lot of real detail, that's not to say that those species resemble each other except for a few tenths of millimeters of a beak. It's just that those characters were easy to use and easy to see under a dissecting scope, so botanists used them. The plants may look quite different, so that it's actually quite easy to, with some practice and some help, somebody pointing them out, to go straight to site recognizing Carex in the field, and you can recognize essentially all of the Carex, even the very difficult groups, on site, walking around without needing to put them under a dissecting microscope. There are a couple of really good sites that have some good Carex illustrations. One of them is the Go Botany site for New England.
Yes. Yep.
It's very nice. And the other is the online Michigan Flora. Both of those differ from the usual online sites in that they also have keys online. And that's much more difficult to do than put up an atlas and pictures.
Right.
So those two, you can generally rely on any of the images that are on there. And there are good websites for the California flora that's very helpful. So between all the various sites, there's a lot of information out there and some good images that you can rely on. But you do have to be very careful.
Well, Tony, today in this series of video clips we've shot, I think for me, we didn't go through 20, 30, 40 Carex, but what we did do, I think, is yes, we did identify these species, but you went through and described these characters and how to even use them in a key, simpler ways. And I see this has been actually a big boost of more field-oriented information, and then of course all the good background information that you've provided. You're just a wealth of knowledge, which I'm sure many have told you, but I would like to thank you for doing this with us, and we really appreciate it.
It's been fun. It's always nice to get wet among my favorite plants.